Welcome everyone to webinar number 5 of this series with Thoroughbred Breeders Victoria and AgriFutures. I'm Justin Darcy, Thoroughbred Breeders Victoria's Media Marketing Officer, and I introduce Annelies McGaw, AgriFutures Thoroughbred Horses Program Manager, Dr. Zamira Gibb from the University of Newcastle, and Dr. Aleona Swegen from the University of Newcastle. AgriFutures do a wonderful job, of course, for our industry, investing into research on issues that affect the whole of agriculture. Now, just a reminder that this session will be recorded, so if anyone is looking to watch a replay or if anyone couldn't make it tonight, it will be seen on both of our YouTube channels. And I just ask for all questions to be held until the end of each presentation, and I'll make sure that they're passed on to our speakers. Now, let's get stuck straight into it. I'll ask Annelies to speak a little bit about what AgriFutures do.
Excellent. Thanks very much, Justin. I really appreciate you doing this for us. So thank you, everybody, for joining me here today. I'm just going to have a bit of a chat about the AgriFutures Thoroughbred Horses program, and then I'm just going to have a brief mention about our Thoroughbred Horses program 5-year plan that is now open for consultation. So I'm going to give a quick overview of the levy and also the plan that is open for consultation now, and then Dr. Aleona Swegen will speak, and then Dr. Zamira Gibb. So it's going to be a great night. So just to quickly go through the Thoroughbred Horses levy is that the $10 from every mare returned is paid by the mare owner, and $10 for every mare served is paid by the stallion owner. And then we invest this levy on behalf of the thoroughbred breeders into research, extension and development. We're also very lucky that these levies also attract a matching amount from the Commonwealth Government, which is 50 cents for every dollar spent on R&D. We're also a really lucky program because we get quite a lot of industry contributions, and we've received industry contributions from the Hunter Valley Equine Research Foundation, from Equestrian Australia, from various vets, Racing Australia. So it's it's great. It's a really good program. We have 24 currently ongoing projects, and some of our key achievements include managing heat stress in stallions, a quicker test for chlamydia and herpes, development of a fertility assessment device for stallions, which we'll talk about today, and also industry support such as sponsoring fast track applicants. And on your screen there, you will see the latest snapshot. So every year the Thoroughbred Horses program develops a research development and extension snapshot, and that is a publication that is on our website, which gives an update on all current and recently finalised research that the program has. So the next thing I want to talk about is the Thoroughbred Horses Program Research Development and Extension Plan that will go from the end of this year until 2027. And what I want to talk about in particular is why do we have a plan, how the plan was developed, and then I'll go into some detail about the different priorities that are in the plan. So we have a plan, and each one of our industries at AgriFutures has a program RD&E plan, and we do that so that we can guide the investment of levies, matching funds and industry contributions into priorities identified by industry through research, development and extension. So the plan commenced development a number of years ago when we did the midterm assessment of the current plan, the 2017 to 2022 plan. We did a midterm assessment of that plan, and then at the end of this plan we did a full-term assessment to understand the economic impact our research was having. Then we conducted in-depth interviews with 26 levy payers to understand what their needs were, and also conducted an online survey for other industry stakeholders to participate in, and a draft plan was then developed. This was then reviewed and edited by the Thoroughbred Horses Program Advisory Panel and has been finalised and is now open for public consultation. Public consultation will go until the 10th of October. And again, we'll put another link in the chat shortly about how you can get to the new plan and how you can provide a comment on the new plan. Once the public consultation is finished, we will incorporate edits, and then it will go to the AgriFutures board for endorsement in December. So these are the 5 priorities that we have listed for the Thoroughbred Horses program. And our overarching goal is to improve the sustainability, productivity and competitiveness of the Australian thoroughbred industry. And I will just go through these in a little bit more detail. So priority 1 is about enhancing the thoroughbred horse welfare, human safety and environmental sustainability. Realistically, welfare is at the core of everything we do with the thoroughbred industry, and so it was very important that this was clearly articulated as our #1 priority for the Thoroughbred Horses program plan. Our main activities around horse welfare will include strategies to reduce injury, breakdown and premature breakdown of horses, also to minimise the impact of climate change on horses, mares, stallions and racehorses as well. And then also research associated with the outcomes of the Thoroughbred Aftercare Welfare Working Group, or the TORG group. When it comes to human injury and illness, we want to look into research to reduce the risks associated with human injury, but also reduce the risk of zoonotic diseases. Then environmental sustainability. This will be about developing baseline data on the thoroughbred industry's impact on the environment, such as our carbon footprint. Priority 2 is around improving thoroughbred horse breeding efficiency. We'll be covering a bit of this tonight, actually, but in particular, our activities will include supporting natural mating and possibly using technology from the human reproductive industries. Then we'll look to improve breeding outcomes and also increase healthy foals, increase the number of live foals and yearlings, including understanding the drivers behind live foal percentages and what it takes to improve them over the next 10 years, improving the understanding of risks associated with delivering a live foal, and then also a possible national fetal post-mortem database. Then with yearlings, we're going to look into pre-sale radiography and also endoscopies and how that can be, what is the actual impact of those and how they can be improved. Priority 3 is around reducing productivity losses and welfare issues caused by disease and parasites. Our activities will include reducing the impact of disease and parasites. This will include foal diseases, diseases and parasites and anthelmintic resistance, and then also antimicrobial resistance. We'll also look at emerging pests and diseases. We have something called the Rapid Response Fund, which is funding available for emerging issues that is quickly available. And then we'll also look at Hendra and other emerging zoonotic diseases. Priority 4 is to increase the industry familiarity with the program, but also to develop a capable workforce. Activities around this will include career opportunities and pathways, supporting industry initiatives such as Fast Track, considering opportunities on how to drive the inclusion of Indigenous Australians within the thoroughbred industry, supporting scholarships, but also training and succession planning for the advisory panel. Within communications and extension, we want to look at implementing the stakeholder needs analysis, which was conducted last year, understanding effective communication of research findings, and then also engaging with industry in such things as like this webinar, but also other events and newsletters. The final priority is around providing up-to-date estimates of socio-economic contributions to allow the industry to demonstrate its value to the Australian economy. And this is all about understanding economic contributions of the thoroughbred breeding industry. And this is a previous version on your screen that we've completed previously, and we'd like to update those numbers. So importantly, this is all available on our website, and I will add the links into the chat after this. And I would appreciate if you could get on there and have a look and provide any comments. So back over to you, Justin. Thank you very much.
Thank you very much for that, Annelies. It was very insightful, those 5 priorities. And I just ask, AgriFutures does do an amazing amount of work. So if you can, please opt into any of their messaging channels. They're very insightful. And again, thank you very much for that, Annelies. But now I'd like to pass it on to Dr. Zamira Gibb. Welcome, and would you be able to tell us a little bit about yourself and what you're talking about tonight?
So my name's Zamira Gibb. I'm from the Priority Research Centre in Reproductive Science at the University of Newcastle. My background is in animal science, and I have a PhD in veterinary science. Most of what I do is for horse industry, horse reproduction work, but I also do a little bit in cattle and humans, and hopefully bees, and have done a bit of oyster work in the past. So tonight, thanks for the opportunity of being able to speak with you tonight. I'm going to be telling you about our project, which is to develop a novel device for the on-farm assessment of stallion sperm fertility. And this is a project that I am doing with my colleague, Dr. Roisin Griffin, who is somewhere out in the wilds of Scone right now on field work. Okay, so just a bit of background as to why this research is important. Obviously, in thoroughbreds, we don't routinely collect semen from them. So this differs from any other breed of horse, which uses artificial insemination. But the reason that this is significant is that because of this, we don't really have a lot of idea about the quality of an ejaculate that's being deposited into the mare's reproductive tract. When you collect semen, you can get an idea of the volume of the semen, the concentration of the sperm, and some idea as to how many potentially fertile sperm are being deposited into that female. So based on artificial insemination research, we know that at least 250 million progressively motile sperm need to be deposited into the female tract for optimum fertility. And if we actually knew how many fertile sperm were in the thoroughbred ejaculate, perhaps we could better manage things like cross covers. So if there weren't enough sperm being deposited into the female at a breeding, then we'd know that that man probably needs to be cross covered before she ovulates. I mean, on top of this, I'm talking about motile sperm, but actually motility is a pretty poor predictor of fertility. Obviously, if sperm isn't motile, it's going to be infertile. But you can have a sample that has pretty good motility and still poor fertility. And this is across all species, so this isn't unique to the horse. So there are other things at play here. And, you know, the reason that fertility is so important is because we need to get our mares pregnant early in the season in the thoroughbred industry, because the industry favours those earlier born horses. So these early pregnancies are more valuable because younger horses are smaller and also slower. So this is just a bit of background, and this probably isn't news to any of you. There's this really great study that came out a few years ago, which showed that each day of age at a weanling sale is worth about $224. So this means that, you know, the youngest and oldest horse at a weanling sale of equivalent pedigree and quality could differ in price up to about $32,000, which is a significant amount of money. And this follows through into later in life. So as horses age, they get faster up until about 5 or 6 years old when it starts to plateau off. And if you do the maths, over an 1,800 metre race, those younger horses, a 2-month age difference can equate to about a 2-second difference in finish time. And obviously that can be the difference between winning and losing that race. So, you know, it's important that the mares are bred and the foals are born as close to the 1st of August as possible, but you all know this. But the way that we can try and do this is to ensure the best chance of pregnancy on every estrus cycle, so that we don't have to wait until the next cycle, and that foal is then going to be, 3 weeks younger than it would potentially be. So in this image here, we have stallion sperm bound to oviduct epithelial cells. So this is in the mare's oviduct. So this is where the sperm will wait for the egg at ovulation. So mares will accept a stallion for about 4 days before they ovulate during that estrus period. And in the wild, because these stallions have relatively small bands of mares, they might breed that mare up to 7 times a day during that receptive period. So those in the wild, those stallions will breed those mares up to 7 times a day. And, so that mare might be bred 28 times before she ovulates. And effectively what this does is it really increases the number of sperm that are sitting there in the oviduct waiting for that egg to come down. Now, obviously, this isn't a luxury that we have for thoroughbred horses who might be expected to impregnate over 200 mares in a 12-week window. And so any cross-covering activities need to be done ideally in a really structured way and only when absolutely necessary. So how can we detect when insufficient numbers of sperm have been deposited into the female after a breeding? To be able to do this, we really need to know what drives fertility in this species. And I'm not going to, time doesn't allow me to get into this, but we spent a good 4 or 5 years basically working out what it was that drove fertility in horses more than anything else. And this is the paper here. So if anyone wants to nerd out, then there's your reference that you can go and check out. But long story short, sperm can produce energy via 2 methods, either via oxidative phosphorylation in the mitochondria in the midpiece, or via glycolysis in the sperm tail. So most species use glycolysis. However, we found that stallion sperm actually depends really heavily on oxidative phosphorylation, and this was one of the major drivers of fertility in this species. So we thought that this would be, a good starting point to develop our device. And the way that we thought that we would do this is using a chemical called rosazurin. So rosazurin is a chemical which changes not only color but also fluorescence via mitochondrial oxidative phosphorylation reactions. So it becomes highly fluorescent and it also becomes like a pinkish color. And this has been around for many, many years. Back in the 1930s, it was identified as a really easy way to identify contamination in milk samples, and it's still used to this day. And then a bit of a deeper dive uncovered a patent from the 1990s where someone patented a take-home kit for analysing your sperm quality using rosazurin. So we thought, well, that could be pretty easy. If we can just add rosazurin to semen and get a good readout of sperm metabolism, then that would be super handy. So can we just mix semen with rosazurin and either have a look at the colour change or measure fluorescence? So to do this, we needed to basically separate out the effect of the sperm cells, which is what we're interested in. Obviously, seminal plasma doesn't fertilize the egg; it's the sperm cell that does that, and the other stuff that's in a dismount sample. So this is just showing a dose response with rosazurin incubated with isolated horse sperm, and you can see that it really nicely does reduce that rosazurin, and we get an increase in fluorescence. But then when we add rosazurin to other stuff that you might be expecting to find in a dismount sample, like blood and obviously seminal plasma, because that's what the sperm are sitting in semen, we find that white blood cells or leukocytes and seminal plasma can actually also really effectively reduce this rosazurin. So that's a problem because we don't want that to be masking the effects on the sperm. So what this told us was 2 things: that the previously published and patented sperm assessment device is really just measuring the ability of white blood cells and seminal plasma to reduce rosazurin, which isn't super helpful. And so for our device, we're going to need to be able to isolate sperm from other contaminants that might be present. So we undertook our first field trial, and during this field trial, dismount samples were collected. We ran some conventional sperm assessments like motility, morphology, and sperm concentration. And then the way that we thought that we would isolate the sperm cells from all the other junk in the dismount sample was to do a swim up assay. So swim up technique rather. So swim up just involved layering culture media on top of the dismount sample, and it allows the sperm to swim up out of that dismount sample and into the culture media, where we can aspirate them off, and then we can incubate them with rosazurin, which then becomes fluorescent, depending, and the degree of fluorescence depends on how many sperm we have and how metabolically active they are. And we can then read that fluorescence using a big clunky piece of lab equipment called a plate reader. So obviously this required a lot of equipment and expertise and time that isn't necessarily available in the shed. But our preliminary results were pretty encouraging. We found that when we separated our samples into those which were dismount samples which were collected from breedings which were successful, so they resulted in pregnancy, or not successful, so they did not result in pregnancy. We had no difference really in morphology or motility, which are sort of the conventional parameters that you'd assess. However, the rosazurin fluorescence was significantly higher in those successful breedings. So this gave us a lot of confidence that we were onto something and to move on. And using that preliminary data, we were able to secure a grant from AgriFutures Australia to produce a device which we coined Equility. And this device, the idea of this is that we can diagnose fertility by measuring oxidative phosphorylation in the sperm. So using rosazurin, obviously, as our reagent. And our first season had really promising results. It showed that, that the fluorescence from dismount samples derived from breedings which were successful was significantly higher than the unsuccessful breedings. So this really just replicated what we'd done previously, but this time we were using an automated device to do those readings. And so this was touted as a speedy test to solve a long-running problem in the thoroughbred industry and attracted quite a lot of attention. And then, of course, we had COVID and we had close downs, and we couldn't travel and we couldn't get parts for our devices. So we had to do a lot of validation work and background research using our research pony stallions. And during this time, it occurred to us that we might have a problem. And that is that, yes, we can measure the quality of those sperm cells, so their ability to undergo oxidative phosphorylation, but still the number that are being deposited is really important to know. And we don't know this because we don't know what the volume of that ejaculate is. So in the words of very famous reproductive biochemist Roger Short, who was talking about dismount samples, he said, you can't tell how many people got on the train based on how many are still standing on the station. And this is a really good analogy. So how are we going to overcome this problem? And this is just an example of what I'm getting at here. So here we have a very small volume but highly concentrated ejaculate. And here we have a very large volume but more dilute ejaculate. However, in both of these samples, we have the same total number of sperm. So they're actually just as fertile as each other. However, if we take a small aliquot from each sample, and this is analogous to a dismount sample, and we don't know how much the volume was originally, well, then that sample on the left is going to look great and the one on the right is going to look rubbish, even though they were actually, the same total number of sperm that were deposited. So we need a way basically of measuring not only the sperm metabolic rate, but also the volume of that ejaculate. And can we measure the volume using our dismount sample? So we know that things like prolonged teasing will increase the ejaculate volume, and that's because we have contributions from all of our accessory glands. And the more teasing, for example, that the horse does, the larger that volume is going to be, and therefore the more dilute your sample will be. And this is really going to cloud what our readings are telling us. So the way that I propose that we overcome this is by measuring a particular enzyme that is only in the epididymis. So the epididymis is where the sperm is stored. So as the sperm becomes more dilute, with a more voluminous ejaculate, so does the concentration of this enzyme. So, and this really nice study that came out quite a few years ago now showed that there was an inverse relationship between the concentration of this enzyme in the ejaculate and ejaculate volume. And preliminary studies that we've done over the last few months with our research pony stallions has shown that the same to be true for the dismount sample. So moving forward, this is what we're going to be using to correct for that, that potentially very dilute sample issue. So we should be able to calculate the number of metabolically active sperm in the ejaculate by taking our equality readout and multiplying that by the inverse of our enzyme concentration. So this should be a much more accurate and more powerful readout. So this is what equality looks like right now. Sorry, I can't work out how to get a laser pointer, but you can see we've got the box of equality. You open the lid, there's a drawer on the side, which is the little image labeled B there. We put our dismount sample in there. We then lower a cartridge into the top of the machine, and I've got them crossed out here because that's where all the magic happens. And this is a potentially patentable element. So in this cartridge is where we can separate the sperm from the other contaminants in the ejaculate, and then the rosacea and reduction assay happens in there as well, and then we get a readout on the screen of the equality device. So the field trial that we're currently involved in, I'm out at Widdenstad now as we speak, in Derek Field's office actually. I don't know if Derek's out there, but they sent me here. I'm not here of my own doing. So we get the dismount samples. We're going to run equality continuously over 1 hour, and that's a typical readout that we see over an hour. And we're going to also collect an aliquot of that dismount sample and freeze it for enzyme quantification. So we're going to take some conventional sperm measurements as well, just to compare them, get our pregnancy results, and then divide those data up into samples which were obtained from breedings which did or did not result in a pregnancy. And then we can use that data to separate our equality readings and work out the best time for the maximum prognostic value. So this is going to look like at the end of the day when this is a commercial product on the bench. Stallion breeds the mare. We get a dismount sample. We run it on equality. If it's all good and the fluorescence is great, well, that mare can then go out into the paddock and have her foal. The stallion can go back to the breeding shed at the next session and cover the next mare. However, if it's a bad reading, then there's still time before that mare ovulates, and if the stallion's book allows it, to rebreed that mare or cross-cover that mare to increase the number of fertile sperm that are waiting for the egg. So with that, I would like to acknowledge our collaborators, the 4 farms that we've worked with over many, many years, Widden, Arrowfield, Darley and Vinery, all under the umbrella of the Hunter Valley Equine Research Centre, the people at the University of Newcastle, so in ANFF and the PRC in reproductive science, and of course our funding partners, so AgriFutures Australia, who've now funded 2 iterations of this project, and all of our background research, which was funded by the Australian Research Council's linkage scheme. And furthermore, with Australian Research Council funding, I just discovered yesterday that I've got a future fellowship, and that future fellowship is going to support me to continue this research for the next 4 years. So thank you very much.
Thank you very much for that. That's an amazing presentation. It just goes to show the amazing work that you guys do and the time it takes to actually come up with a product like this. It's incredible. But if anyone does have any questions, please feel free to ask them in the chat now. Otherwise, we will go straight over to Dr. Aleona Swegen. If anybody does have any questions for our last presentation, please feel free to put them in the chat and I can go back and ask them at the end. But without further ado, I'll introduce Dr. Aleona Swegen. Thank you.
Hi, everyone. Thank you for the introduction and thank you for the opportunity to give an update on our project. I assume you can see the presentation up there?
Yep, sure can.
Great. So I work quite closely with Zamira at the University of Newcastle. I'm a researcher here as well, and this is a somewhat so my background is in veterinary practice, and I worked in practice for a couple of years before I got seduced by the wonders of research, and also really interested in male fertility. But one of the the things that we've started to introduce into our research program is the female side of things and early pregnancy in the mare, because this seems to be one of the under-researched areas where we do have a lot of questions about what's going on. And so this is a somewhat younger project and was actually first really funded ever by AgriFutures Australia through the Thoroughbred Horses program. So very excited to get this off the ground, and we've been working on this for the last couple of years. So there's a couple of aspects to this, but by way of background, there was a study a few years ago that basically showed that if we look at mares at day 2 post-ovulation, of the mares that have been bred, actually about 90% are successfully conceiving. But by the time we scan them, which typically happens at day 14, as I'm sure you know, we're only seeing a pregnancy rate of about 65%. So what this means is that about 10% of mares are not conceiving, but then another sort of 25 look like they are losing their embryos quite early on before we even get to scan them. But ultimately, those two things combined mean that about 1/3 to 1/2 of mares after each breeding cycle will scan in negative, and they'll return to be bred again. And also what we learned from this is that the risk of early embryo loss is quite high, and we don't really understand why. So Zamira talked quite a lot about this. This is the same study that I will reference, where we know that the age of weanlings when they go to sale is strongly correlated with the hammer price. And basically, a breeding operation is suffering economically every day that a mare isn't getting pregnant. So timing is really important. We've got a really short breeding season, and even if we're not talking sale prices, of course, physiologically there is that limited window of opportunity for us to breed these mares. So really the idea behind this is to be getting mares pregnant earlier. So typically what happens is, of course, we have our ultrasound at day 14, and if that mare scans in negative, well, then she can't be bred until the next estrus. What might happen if we had a pregnancy test at day 7 is that you could short cycle that mare and then rebreed her just a few days earlier, but it all adds up over the season. And especially if you think about mares that come back multiple times, if each cycle is that little bit shorter, she might have an extra opportunity by the end of that season to go and foal rather than be left barren until the next season. So all of this would have economic benefits. And the other side of this is really understanding the biology of why and how embryo loss happens, because that's still something we know quite little about. And if we can understand some of those mechanisms, we're hoping that further down the track, we might be able to come up with some treatments to address this major cause of breeding inefficiency. So the priority objective that AgriFutures had identified in the last round of our DNA priorities that this project falls under is the research leading to the early diagnosis of pregnancy in mares. And there were 2 components, like I mentioned. So the first is working on an early pregnancy test, and the way that we are doing this is by collecting bloods from pregnant and non-pregnant mares and really comparing various aspects of the biochemistry in those samples, so that we can then use that knowledge to hopefully develop something that might look like a nice and simple test for early pregnancy. And the second component is really trying to understand more about the interaction between the embryo and the endometrium of the mare, because there's a lot of communication that goes on between the two that we don't know much about, and then hopefully use this to develop treatment. So ultimately, we are hoping that we'll attain a shorter foaling interval and reduced embryo loss. So on the pregnancy test side of the project, it's a fairly simple, conceptually experimental design. So we're working with our collaborating studs in the Hunter Valley, and we're collecting plasma samples at day 7 and day 14. And then we're taking advantage of some mass spectrometry technology that we have access to here, which is a really great opportunity, I suppose, in that this kind of technology is rarely made available to people like us in the veterinary and agricultural space. It's often reserved for biomedical sort of applications, and it's really nice that we get to access it. And what this can do is analyze these samples at a really, really high resolution and give us a lot of information that we previously haven't been able to get in the more basic kind of biochemistry tests. And we can learn a lot about almost all of the proteins and the lipids and the metabolic sort of components that are floating around in the blood of the mare that we might be able to use to distinguish pregnant and non-pregnant animals. Then the next step after doing a kind of screening component is really identifying some candidate proteins or lipids or metabolites, and then validating these to make sure that the biology underlying this is really solid before we go on to design a prototype test using these candidate biomarkers that then can evolve in future research into something very practical that we can use on farm. And looking at embryo-maternal interactions, the approach here is recovering embryos from mares generated in vivo, and then essentially incubating these in a dish with something called endometrial organoids. And what these are kind of little blobs of cells that are cultured in 3D, and they're able to mimic a lot of the functions of the endometrium. So they're quite similar structurally. They have glands. They release some of the same components that the endometrium does. And so we can really get a closer look at the biological interactions that happen between the embryo and how the embryo signals its presence and what can kind of go wrong during that process. And get a really close look at this by doing these experiments in the lab. And also then look at what this model of the endometrium does in response, because the hypothesis is that some of these things don't always go to plan, and we're hoping to find out more about this, and we suspect that some of these things are leading to early embryo loss in the mare. So I'll focus mostly on the pregnancy test component today, but this is the sort of timeline of the project, which is coming into its third year now. So we've collected the bloods and we've identified a series of biomarkers, and we're now refining and validating those biomarkers before we hopefully generate a sort of prototype before the completion of the project. And on the embryo-maternal interaction side, we've got all our tissues, and we're wrapping up the final in vitro experiments before we do the analysis on those. And so the first part of the screening component, so the early pregnancy test project, was looking at proteins using a proteomic platform. And so we've looked at bloods of day 7 and day 14. Of course, the day 7 ones are the exciting ones that we're interested in. And so this is what's called a volcano plot, and essentially there's often a kind of overload of data, but what we want to see are proteins that are coming up both as statistically significant, so they're consistent, so they're towards the top of the chart, and the ones closer to the outside on the horizontal axis are the ones that are kind of most dramatically up or down regulated in the pregnant versus the non-pregnant animals. And so we're seeing quite a few that fit that profile, and some that are more consistent than others. So one of the proteins in the serpin family up here have come up as quite consistently different between pregnant and non-pregnant animals. So this is pretty exciting, given that for a long time it was thought that there are no real changes in the circulation of the mare in response to pregnancy until day 14, or at best day 10 following ovulation. So we are seeing that there are some physiological changes that can help us pick up those pregnancies quite early on. So overall, there were 9 proteins that we considered statistically significant significantly different between pregnant and non-pregnant animals. But of course, we want those to be really consistently different before we can go forward with those as a pregnancy test. And what we're thinking is going to happen, so this is just another way of visualizing it, that basically, so we've got pregnant horses on this side and the non-pregnant ones here. And the red are up-regulated and the green are down-regulated proteins. So we can see that some go up and some go down. in response to pregnancy. But what we're thinking is that we're going to need a panel of 2 or 3 or 4 proteins that we can combine together to give us a robust test. And so when we do this, we combine them into a formula. We get a pretty good separation of the pregnant and non-pregnant animals if we look back on our current data set. So what we can see is that if we draw a line at a sort of theoretical cutoff here, we can see that the pregnant animals all cluster quite closely below that line. So this is really important that we're not getting any pregnant animals showing up in the non-pregnant space, because we don't want to be accidentally picking up pregnant animals as non-pregnant. This has pretty dire consequences. But most of the non-pregnant animals will come up above the threshold, and then we've got what we're calling our false positives down here. And this is not ideal, but also it is to be expected, partly because if we're collecting these bloods at day 7 and then confirming animals as pregnant or non-pregnant at day 14, we do expect to be picking up some that were pregnant at day 7 that have since lost their embryos, and therefore they're scanning in as non-pregnant at day 14. So some of this is an expected result. So this is a sort of example of where this pregnancy test is headed. So we're going to use a combination of factors in the plasma that we can then take forward for a test. So that's just the proteins. Then we're also conducting a lipidomic study. This is still underway, and this is a similar sort of volcano plot. Unfortunately, lipids are much more poorly characterized than proteins, so we don't have nice names for them, and we also don't have as much information about what they do. But we're certainly seeing some that are quite dramatically up and down regulated in response to pregnancy, and this is still ongoing. And then on top of that, we're also doing a similar sort of screen for metabolomics, so some of the small molecules floating around in the blood that may also differ between pregnant and non-pregnant animals. So this is just a very brief summary of where we're at and what we're currently doing for both the proteomics and the lipidomics is moving on to some more targeted techniques where we can really make sure that those changes are consistent across a large data set of animals. And then we're going to move forward and design a device that, at least in theory, we can take forward into further projects. As you will have observed from Zamira's presentation, the device design and optimization component of these kind of studies is a whole story in itself. So I just want to introduce you to the people that have been doing the work, most of the work that was presented in that talk. So Rukmali here on the left has been has really taken the lead on the pregnancy test project and has been very laboriously working through these huge data sets and processing hundreds of samples. Steph has been working on the endometrial organoid project, and Alicia has been the research technician that has made it all possible for us as well. And of course, thanking AgriFutures for the funding and the really generous support of the 4 studs that we've worked with through the Hunter Valley Research Centre as well, and some of the other people in the team at the Priority Research Centre for Reproductive Science here at Newcastle. So that has been a very brief update, but I'm happy to answer any questions that anybody might have. So I will leave it there. And thank you.
And just before we go, we actually have a question.
So do we? Beautiful.
Oh, we do. It's from Dr. Catherine Chicken, our advisory panel chair. So Catherine, you had a question.
Ali, Zamira, thank you very much for your presentation. So great. Ali, do you envisage that you will have a combination of proteins and lipids as the, in the test? Or what are you anticipating from what you've seen so far?
So I am hopeful for a multiple protein panel. I think where we are at in the wider scientific space with detecting proteins, it's a more linear, it's a more straightforward process, but it could still be a combination of all of them. I just think that is going to be more challenging to design. I'm hopeful we can get away with proteins. The lipids are showing up some really interesting biology as well. So I don't want to give up on them.
Excellent, thank you.
Thank you very much, Annelies. Could I ask Zamira, how consistent is the OXPHOS level on a ejaculate to ejaculate basis, Zamira, per stallion, as it were?
Yeah, well, we are. So I'm right now collecting that data, but I haven't actually analysed it. So the previous runs, we basically got data from every stallion, but only a few, 2 or 3 ejaculates, and it was quite variable in that case. So this time round, we're focusing on just 4 stallions, so that we can get a lot stronger, larger sort of sample sizes within that stallion. What I'm seeing is just what I'm seeing on the screen, so I can't give you anything other than sort of a qualitative answer, and that is that I see very similar patterns for within the stallion. I've only been doing it now for a week and a half, so I think that their ejaculate quality probably won't have changed that much in that period of time. But interesting things like if there's a lot of gel in the sample, it will take a lot longer for the fluorescence to start to increase, because I think it takes a bit more time for the sperm to actually escape that gelatinous seminal plasma. And, obviously stallions who are, we call them jelly monsters, tend to have a gelatinous ejaculate, most of the time. It's not sort of a hit and miss thing. So, yeah, long story short, yes, they seem to be fairly consistent patterns, but that's just within a small time frame. So, I think that the magnitude of the fluorescence intensity will differ a lot more over time.
Okay, thank you.